Okay. Are we live now? Tanya, can we just confirm to me that we are already?
Yes.
Yeah. Okay, so we just wait a couple of more minutes for people to join. Hello, everyone. Okay, maybe we can start. So good morning or good afternoon or good evening, everyone, depending on where you are watching right now. It is my pleasure to welcome our speakers and all of you to this session of the Marine Biodiversity Networking Fridays organized by the Air Center. This session is part of the GOOS Bioeco Essential Ocean Variables webinar series. And like the previous session, it is being live streamed from YouTube, from the Air Center channel, and through a Zoom session for those who registered. My name is Ana Mafalda Correia, and I will be moderating this session. I am a marine biologist, a postdoc at the Interdisciplinary Center for Marine and Environmental Research at the University of Porto, Portugal, and I have been dedicated my scientific career to station ecology and conservation. And I would like to start off by thanking the Air Center for inviting me to moderate this session, which will be dedicated to seabirds and marine mammals abundance and distribution. As some of you may know, these marine megafauna species are key to the functioning and structure of our ecosystems and important sentinels for the ocean's health. And throughout this session, we will be diving into the world of these magnificent organisms and hear about why seabird and marine mammal data are vital for healthy oceans and smart decision making, the current status of ocean monitoring and gaps needing attention, and how to amplify your impact via the GOOS International Standards for Ocean Observations. Within that line, you will learn also about meaningful ways that we can all contribute to ocean monitoring. So during this session, we will have three speakers with us, and I will introduce them now. We will start with Dr. Samantha Simmons, who will introduce us to GOOS and the essential ocean variables. Samantha is a marine mammal biologist focusing her research on seals and sea lions with a PhD from the UC Santa Cruz in California. After 13 years serving as an advisor on science-based policy to the US Marine Mammal Commission, she then returned to her origins in the United Kingdom to be principal scientist at the SMRU Consulting. She has been a panel co-chair for GOOS Bioeco and is now representing the marine mammal essential ocean variable on the panel. Following Samantha, we will have Dr. Patricia Serafini, who will guide us through the seabird essential ocean variable. Patricia is a veterinarian and currently a PhD candidate at the Federal University of Santa Catarina in collaboration with the University of Oxford and the British Antarctic Survey. She works on the conservation of albatrosses, petrels, and other seabirds since 2010 and serves as an environmental analyst at the National Center for Research and Conservation of Wild Birds. Patricia is a co-convener of the Population and Conservation Status Working Group of the Agreement for the Conservation of Albatrosses and Petrels. Finally, last but surely not least, we will welcome Dr. Dipani Sutaria, who will talk to us about the marine mammal essential ocean variable. Dipani is an environmentalist and ecologist with a PhD in Earth and Environmental Sciences from the James Cook University in Australia. Since 1998, Dipani has been studying marine megafauna, including turtles, elasmobranchs, and marine mammals in India. She's an adjunct senior researcher fellow at the JCU Australia, research affiliate at the Dakshin Foundation India, and a member of the IUCN Station Specialist Group, the Marine Mammal Protected Areas Task Force, and is chairing the Conservation Committee at the Society of Marine Mammalogy. In India, she is part of the Marine Mammal Research and Conservation Network and has played a key role in the designation of important marine mammal areas in the Arabian Sea and the Bay of Bengal. So about this session, we will have these three lectures. each one with about 10 to 15 minutes. And then we will have a 15-minute open discussion, and registered participants can ask their questions by writing them under the Q&A box of the Zoom session. Please, not in the chat. Try to write them on the Q&A box. And then I will put the questions to the speakers, and I will try to moderate these questions in the order they appear in the Q&A box and the speakers they are addressed to, and try to manage the time that we have available for discussion. And I think I have nothing else to say. So without further ado, let's move on to the first speaker. Samantha, please, the floor is yours. You can start now. Thank you.
Wonderful. Thanks very much, Anna Mafalda, and thanks everybody for joining today and welcome. As you just heard, I'm going to introduce you to the Global Ocean Observing System and the essential ocean variables at a high level, and then you'll hear more from Patricia about seabirds and Dipani about marine mammals. So I think we've advanced slides, and GOOS was formed in 1991 in response to calls from the Second World Climate Conference in Geneva the previous year. Over the last 30 years, GOOS has connected numerous observing networks, platforms, and monitoring programs, so that today, and I do mean today because the 2025 GOOS status report was actually released yesterday, so this is hot off the information in terms of our new metrics. We have 64 countries contributing observations to the global system. That includes 17 mature ocean observing networks, including drifting buoys, tide gauges, Argo floats, HF radar, ocean gliders, and animal-borne sensors, as well as several emerging networks. There are 12 active biology and ecosystem communities, and throughout the talk today, if you hear myself or Patricia or Dipani say bioeco, that is just us squishing biology and ecosystems together. So we have 12 active bioeco communities, and every day all of these networks are taking a total of more than 120,000 observations feeding into the global system. There are also more than 600 long-term active biological monitoring programs that are collecting marine biological and ecological data. And all of this is to realize the vision of a truly global ocean observing system that's delivering essential information needed for our sustainable development, safety, well-being, and prosperity. The vision to deliver this essential ocean information is realized by having coordinated observations that can be integrated, are open and available, and fit for purpose, with the potential for multiple uses at different scales. So we're really going with the mantra of measure or observe once and use that measurement many times, where the information and products developed have real impact and applications for society and the sustainable use of our ocean. GOOS does this by focusing on 3 delivery areas. And across these 3 areas, again, GOOS is aiming to provide ocean data with an impact. The areas are climate, operational services, and ocean health. And for the bioeco essential ocean variables, we're feeding mainly into the ocean health delivery area, but also with some impact and input into the climate area. So how do we start on this journey? The backbone of the observing system is the essential ocean variables, and these currently span 3 disciplines: physics, biogeochemistry, and biology. On the slide, you can see a total of 13 physics EOVs in the dark blue, 8 biogeochemistry EOVs, the orange circles or icons, and 12 bioeco EOVs in green, plus 3 that are cross-disciplinary. More information on all of these is available at the link in the bottom right-hand corner of the slide, and those EOVs with an asterisk next to them are also essential climate variables recognized by the Global Climate Observing System, or GCOS. By using this essential ocean variable framework as a common language and standardized approach to collect ocean observations across disciplines, researchers, policymakers, and stakeholders can collaborate more effectively, leading to comparable observations for better understanding of ocean processes and informed decision-making to manage and protect marine environments. So looking at the biology and ecosystem EOVs, these EOVs were identified to help understand and forecast marine life. And if you're interested in the process to identify the initial suite of EOVs, you can read more about it in the publication in the bottom right-hand corner of the slide. Of course, today we're focused on the seabird and marine mammal essential ocean variables, but like the others, they provide a framework for coordinating ocean observations, ensuring globally comparable and combinable data. How do we get to that globally comparable and combinable data? Well, there are several different pieces to the puzzle that I'll talk about in the rest of my presentation. But the first key one is that for each essential ocean variable across all the disciplines, we have a specification sheet with common terms and ways of defining the observations that are of interest to GOOS. That's not advancing. There we go, sorry. The specification sheets provide a common framework for coordination and guiding collection of ocean observations. This, combined with data standardization, are key to being able to realize our measure once, use many times to deliver ocean data with impact. So I'm now going to introduce you to some of the key terms in the specification sheets, provide a little context for them, and then you'll hear more about them in detail for seabirds and marine mammals from Patricia and Dipani. To specify the essential ocean variables, we go from subvariables to derived products that allow us to understand phenomena and deliver to GOOS. So I'm going to walk you through that kind of process and flow from observations to products and define these terms a little bit better now. Starting with our essential ocean variables, the essential ocean variables are where we start, but to obtain these, there may be other key measurements that are used to estimate them. So these are called subvariables. And for example, for me as a seal biologist originally, if I want to understand or estimate abundance, how many seals are there, I might go out and count individual seals on the beach. So that count data is our subvariable. In addition to the subvariable, we have other measurements that are useful to provide scale or context to the subvariable. These are known as supporting variables. And for my example, with counting seals on the beach, the ocean or air temperature or the tide height might influence the number of seals that are hauled out and available for me to count. Those would be the supporting variables that I want to make observations of as well. This pyramid of sub and supporting variables, along with the EOV, is the foundation of the specification sheets and observations that are important to GOOS. But in addition to that, we want to get ocean information with impact, the why do we care? So in the specification sheets, we also identify phenomena and derived products. By that, we mean the phenomena are example events or processes that can be observed by measuring the essential ocean variable. In the case of seabirds or marine mammals, that's monitoring population status and trends or looking for shifts in distribution. The derived products are outputs calculated from the essential ocean variable, often in combination with the supporting variables. These contribute to evaluating change in phenomena and delivering impactful ocean information into GOOS. So again, you're going to hear more about the details of the specification sheets for seabirds and marine mammals from Patricia and Dipani shortly, but there are three other resources that I want to mention first that are key to this delivery of globally combinable and comparable data. That is starting with data collection, analysis, and standard operating procedures or best practices for our foundational sub and supporting variables. The Ocean Best Practices System is a global initiative with the vision of having and sharing broadly adopted methods across ocean research, operations, and applications. There is a process in the Ocean Best Practice System for GOOS to be able to endorse practices that we recognize as best for the collection and analysis of EOVs, but more broadly, this is a really valuable resource for the community as a living repository for operating procedures and practices. So if you're not familiar with the OceanOps Best Practice System, I encourage you to go check it out. The second resource I want to touch on is the GOOS BioEco data portal, which is available at bioeco.goosocean.org. And this tells us who is currently collecting what data and where. GOOS is interested to have a record for all active data collection, but especially those that have more than 5 years worth of data or have longer term intermittent data collection, such as the scan surveys for marine mammals in European waters. So please go check out the portal. And if your organization is missing and you're collecting those type of data, 5 years worth, feeding into the EOVs, you can use the QR code or the link in orange on the slide to create an entry for your institution or organization. We can extract the data or metadata for storytelling purposes regarding the state of global ocean observation. We're going to take a quick look at what the portal looks like for seabirds and marine mammals. But before that, I'll just mention the portal is currently undergoing a revamping process. And so if you go to the link bioeco.goosocean.org. It doesn't look exactly like what I'm about to show you. Don't worry, you're probably still in the right place. So I went to the portal yesterday or the day before, and in the top left-hand corner here, you can see you can filter by EOV. So I've put on the filter for birds, and we have 108 monitoring programs. You can see relatively good spatial coverage, but there are some obvious gaps. And looking at the same thing for marine mammals, we now have 184 monitoring programs, slightly better coverage, but there's still room for improvement. And again, Dipani and I are aware there are observing communities or programs that are not currently represented in the portal. So if you're listening and you can help fill some of the gaps, please visit that website at the QR code and get yourselves on the map. The information in the portal also provides points of contact for us to engage with when we're thinking of refining the specification sheets or identifying and endorsing best practices, for example. And even if the data you're collecting are not yet in OBIS, there's value in having your institution or organization or efforts registered in the portal so that we know what is being collected, what data are being collected and where. And the third and final resource I'll mention, which is perhaps the most important, is of course the data themselves. And for GOOS, this is with OBIS, the Ocean Biodiversity Information System at obis.org. This is the world's largest open access repository for standardized marine biodiversity data. When originally developed, it was just for species occurrence data, but it's developed exponentially over the last 10 or 15 years in particular to now be able to handle many different types of data, DNA-derived data, satellite tracking, image-derived data, habitat ecosystem, and abundance data. So hopefully I've given you a broad overview of GOOS and the essential ocean variables, pointed you to a few key resources that allow us to ensure we're delivering globally combinable and comparable data. And now I'm going to turn it over to Patricia and Dipani to tell us more about why it's important to monitor seabirds and marine mammals, the details of the specification sheets for those EOVs, and the types of data and information GOOS is interested in. Patricia.
Thank you very much, Sam. Just please confirm if you can see my screen.
I can.
Okay, perfect. So thank you so much. I'm really glad to be here. Thank you for everyone that's here with us today to listen a little bit more about the GOOS and the bioeco and our EOVs. I'm here just a messenger because there are a lot of people that have been worked in the seabirds abundance and distribution essential ocean variable. And Tammy Davis and I and myself right now are the leads of the seabird EOV under GOOS. But we do have a collaboration of a lot of people from the seabird expert community. So I'm here just to share things that we have learned and to learn from you guys as well. So the seabird abundance and distribution essential ocean variable is it's really informing a lot of process on conservation process and sustainable development goals from the United Nations process. And there are several reasons to measure seabirds amongst the minimum set of ocean variables to assess the global health status of the ocean, which made them an essential ocean variable. But there are also many ways of doing so. That's why we have been intensively working with the seabird expert community in a specification sheet that Sam kindly explained to us how it works. And that is an ongoing document bringing guidance and advice on how to better integrate observing programs on seabirds to a global broader view on GOOS, considering standards and accepted methods. But before we dive further on the specification sheet content and its importance to understand these reasons to measure seabirds, I will share a little bit why to measure seabirds. Seabirds are very important indicators of trophic system health because they are top predators of marine food chains, and also they're really long-lived vertebrates. Here in my slide, you can see Wisdom, which is really famous seabird lady, which is already 75 years old, just got back for her colony in the Pacific. And last year, with 74, she had a nestling, which is in the photograph shared broadly and within the community and abroad. So it's long-lived top predators, seabirds, that as top predators are indicators of prey presence and make also very important cues to fisheries, for example, as conspicuous being in the sea. Additionally, seabirds are an important connection between two worlds, the subaquatic where they feed and the islands where they breed. They breed in colonies within land, and they bring all the nutrients from the sea to this land that wouldn't be there otherwise. So they're really key to nutrient dispersal, enhancing primary productivity in this environment, and offering ecosystem services on islands and other coastal areas, making it very important for the human dimension of the ecosystem service they bring. For example, here there's a paper that was published in Science Advances recently showing how important seabirds nutrient dispersal is even for corals around the islands where they breed. And considering that they are also important indicators in biogeochemical oceanography and climate change, and considering that EOVs GOOS are ocean variables that need to have high feasibility, low cost, replicability, and impact to make possible to understand what's going on within the ocean, we can say that we can find all that in the seabird essential ocean variable. The seabirds are thus vital indicators of ocean health due to their mobility and reliance on diverse marine ecosystems, because they are present in every ocean on Earth. They're really like broad from Arctic to the Antarctic, every ocean have seabirds. In this context, we do have 3 sub-variables that have been defined for seabirds, for the seabird EOV. We have the first sub-variable is the species population census or count data, the number of mature individuals of a species. And the second one is the repeated individual presence, obtained through tracking or resides during ocean observation, and to have a cue on spatial distribution. And also we do have as a third sub-variables mortality events for this group. As supporting variables for this group, we do have some EOV related, which is breeding success for the species, diet, behavior, and other demographic threats as age, sex, and others. And also environmental supporting variables that are shared with other EOVs as well, as temperature, salinity, surface and subsurface from the ocean, dissolved oxygen, suspended particles, ocean color, primary and secondary productivity, prey availability, among others. And so variables are supporting variables, as Sam explained to us, are other measurements that are useful to provide scale or context to the sub-variables of the EOV or in providing the derived products to better understand what's going on with the sub-variables we chose for the EOV. So here we do have one of the examples of breeding success that we can access within the seabird EOV. But what we want to observe with the sub-variables that we chose, so we have 3 sub-variables. What are the phenomena to observe? So now the phenomena to observe first is the population status and trends of seabirds. Here I bring an example of effort that we have been engaged in the agreement for the conservation of albatross and petrels, ACAP, and there is a intersessional group on working with population status and trends for albatross and petrel species on ACAP, and led by Johannes Fischer from New Zealand. Here you can find this document and preliminary results on his ResearchGate website. But it's an example of how important it is to have count data of seabirds in colonies and to be able to establish population status and trends for the seabird species we have. Another phenomenon to observe is distribution shift of seabirds species. It's how the seabirds are using the ocean and how it has been changed within time, in a time scale that we can define. It's really important to indicate what's going on climate change, prey availability, and every other causes of ocean change and health. And for example, we do have this paper, just an example, that was studying the interdecadal shifts in seabird distributions. So it's one of the products that we can aim more globally with this global view that GOOS can bring, having data globally shared and distributed. And the third phenomenon to observe that we have for the seabird EOVs, mass mortality events. Once we do have a subvariable on mortality, when we do have a baseline on mortality on colonies or even in non-breeding areas, it's easier for us to be able to identify when we do have some abnormalities and some non-normal situations, as for example, the panzotic that we have been able to see since 2022 in many seabird colonies with a lot of mortality, mass mortality. So it's able, when we do have the information along a timeline and globally, we can understand better the health of the seabirds and the health of the ocean in many, many ways, including the diseases and panzotic as the avian influenza, high pathogenicity avian influenza. If you do want more details on how it has been affected, for example, the endangered albatross and petrels under the ACAP agreement, we do have a webpage where we share updated maps on occurrence and where the disease has been identified and which are the mortality that have been caused. So it's one of the other potential phenomena that we can increase observations in the ocean and understand better what's going on. And for observing requirements for this phenomena and for the subvariables, each subvariable there can be measuring 3 levels, options for the observing efforts around the globe for collecting observations. Minimum one is the minimum required to ensure that data are useful to observe the phenomenon. The second one is desirable. Additional above minimum that can enable additional use, for example, may provide the ability to capture other. Sorry, can you hear me?
Okay, now yes. I was not sure if it was my computer or
Sorry.
your internet, but now I think it's working again. So if you want to share again your screen, but it stopped. It interrupted for
Can you just tell me on which slide I was interrupted and I lost connection? Sorry.
Yeah, you can share and I will tell you. Can you share your presentation?
Okay.
Okay. Wait. The previous one. It was between the previous one and this one. Yeah. It's so maybe you want to start
here.
Yeah, I would say
the phenomena to observe.
Okay, sorry for that. To get a bit, yeah, no problem.
Yeah, I think it was my connection here. Sorry for that. The third and last phenomena to observe that we define with the subvariables that we have is mass mortality events. If you do have a mortality subvariable that we can have a baseline for that colony or for that non-breeding area, it's easier to identify when we do have abnormalities and something not going on right in the ocean. Can be related to disease, to pollution, to other causes and fisheries impacts and other causes. So when we do monitor the mortality events, we are able to identify, for example, the mass mortality events caused by a panzotic, as we have been witnessed with the avian influenza, the high pathogenicity avian influenza, that has been caused several mass mortality events around the globe on seabird colonies. So when we do have information on what is expected on mortality, we can identify ocean health problems along the time series. So I think for an example, we do have for albatross and petrels from the agreement on the conservation of albatross and petrels, we do have a website where we have been for 3 years now shared the information on mortality on albatross and petrel species around the globe So it's really important to have this for all seabird species in a more globally way to be able to identify ocean health in a broader scale. And when we talk about subvariables, there's some observing requirements that we can try to assess on collecting observations, on observations networks. There are 3 levels of options of observing requirements. The minimum, which is the minimum required to ensure that data are useful to observe the phenomenon. The desirable, additional above minimal, that can enable additional use. For example, may provide ability to capture other phenomena or may improve understanding of the phenomena that we have established. And the ideal, above which further improvements are not necessary or will not provide any additional insights. And in that case, I will just go to each of the phenomena and EOV subvariable and talk a little bit about information on observing specifications or requirements. For the phenomena population and status and trends and the subvariable species population census or count data, the idea would be every local colony level or monitored on every breeding season or monthly in tropical seabird species that breed annually. And the desirable would be every colony level having count data at an annual frequency, and the minimal 1 colony level for the species every 10 years. Of course, that standardized specification on uncertainty and effort will be important for comparison along temporal scales on count data at the colony levels, and different methods will result in different metrics, and this will need to be considered and specified, especially to compare along timelines within colonies. There are several methodologies that have been discussed and established along the way. So we do have, for example, books on that since the 90s. And the Albatross and Petrels Agreement, ACAP, also have guidelines on census methodologies for albatross and petrels that can be extrapolated for other seabird species. And there are a lot of literature on census or count data for seabirds. So there are lots of reference. And the most important proxy is nest counting, and then to extrapolate that for mature individuals of that species in the colony. And the second phenomena to observe, which is distribution shifts, it's measured by the subvariable repeated individual presence to tracking or resites at sea. And the ideal situation would have every colony and level population monitored to tracking or to resites at sea, and in every breeding season. That would be ideal. The desirable would be a regional effort annually, bringing information on distribution of seabirds at sea, and the minimum regional, but every 5 years, bring information on distribution at sea of seabirds. Of course, distribution shifts need to take changes in tracking technology over time into account, and there are some very technical details that can be discussed. But and for example, we do have data on seabird distribution at sea shared online broadly on seabird tracking database and other platforms. So just for an example for this subvariable and observation specification and requirements. As the third phenomena to observe is the mass mortality with the subvariable absence of dead seabirds. And that ideally could be measured by visual count of carcasses at a local level monthly. We do that in Brazil. I just brought an example. There are other places in the world that do that as well. Here, Simba is an online monitoring system that shares information that is acquired on a beach monitoring service for carcasses that have been washed up on shore. And that is made daily for some areas in Brazil. At least 30% of our coast is measured every day for strandings of seabirds, turtles, and cetaceans and marine mammals. So we do have a lot of information for that regarding visual count of carcasses at a local level. So it's not monthly, it's daily, but monthly would be the ideal. And desirable would be the visual count of carcasses at a regional level, regional compilation information monthly or annually, and the minimum, the visual count of carcasses in one reference location annually. Of course, we do need to establish a tier threshold to understand what is mass mortality and what is just baseline mortality. For example, in a colony as well, when we do register mortality, we can have every year a natural mortality, but when that is above 0.5% of the population or above 1,000 individuals in one colony, that can be considered mass mortality, as we have been discussed with experts. And this would also allow for retrospective creation of time series. So just one other example of subvariables and observation specifications or requirements we can have. From these subvariables, we can have the potential derived products to understand the phenomena and deliver to GOOS a more global and perspective of the EOV. So for seabirds, we can have as derived products, for example, the population estimates, including total number of mature individuals per colony or island group. And that will inform, of course, the process that's a little bit more complex to have, but we can have information about population status and trends. It's increasing, is decreasing, is it stable? And that will help the red list discussion on status change for seabirds. We can also establish hotspots for conservation, hotspots for threats, and home range, migration pathways, habitat maps, distribution shifts and patterns of habitat use, and of course the documentation of mass mortality events. For example, for the most recent high pathogenicity avian influenza that has increased the number of seabirds mortality around the globe. The requirements for observing networks to be able to share data on GOOS have been already shared by Sam in the beginning of our webinar, but the mission of these observing networks has been, they need to be fit for purpose to be able to share their data openly to support science policy and management needs. That's the most important thing that we can do with our data in the end of the day. And in a spatial scale that it's relevant to bring indicators globally. And of course, we have to try to have repeatability within every local that we monitor and we observe to be able to have a time series that's relevant to understand phenomena, and in a global accepted standards to have the best practice on our observing systems. And of course, using fair data standards of, for example, open data, metadata supporting interoperability, data aggregation, and reuse, and capacity development and technology transfer. That's what some of the ideal requirements of observing network. And I just bring here finally some examples of existing observing networks for seabirds. One of the examples that I mentioned is the monitoring of seabirds stranding networks in Brazil, and also other vertebrates stranding in all around the globe. They're just an example. The agreement on conservation of albatross and petrels as well, bring information on count data on colonies every year, trying to bring the most updated information on population numbers on colonies and island groups. And there are several other initiatives of those, as the Australian Antarctic Program, the Arctic Marine Biodiversity Observation Network, CPOP, BirdLife International has also a lot of experience to share on tracking data, for example, and others. So I guess that was a quick overview. Sorry for the interruption in the middle, but I understand that at least we can have an idea of the EOV seabirds and how it can deliver information to GOOS and broadly. Thank you so much for your attention.
If you can stop sharing, Patricia, I can
Yeah,
start
thank
again.
you, Patricia. We'll move
Sorry,
on to
I thought I
Deepani.
did it. Thank you.
Okay.
Okay, Deepani.
Thank you. Thank you everyone for being here. Morning, afternoon, I don't know from wherever you all have signed in. And as Sam and Patricia have already covered a lot of ground over here, I'll stick to what I love most, which is marine mammals. I won't go into all the details of the technical aspects. So why has marine mammal has the marine mammal EOV been decided, to be included in the GOOS process? Next slide. So, yeah. So, as many of you might already know, marine mammals are a vast group of animals. They have come to the sea from convergent evolution from different lineages from land. So there are there are cetaceans basically that have odontocetes and mysticetes. They're toothed whales and baleen whales, so they have different prey preferences. Then we have the sirenians, the sea cows, and the manatees. They have a different food prey preference, kind of food they eat. And then we have the pinnipeds that we have very few of, actually. We don't have none in my part of the world, and I don't know much about them, but they use both land and sea. Not only that much, they also have different habitat preferences. So we have these animals in rivers, freshwater systems, we have them in lagoons and estuaries, we have them in coastal areas, we have them in seagrass beds. So these animals have gone back from land to sea and evolved in a diverse varieties of ways to live in the water column. So other than that, that's just a basic background of these animals and their niche partitioning that is already available, that is already seen. They are very, very important in the marine ecosystem because they are top predators. So they manage the food web and the trophic system health. They are also responsible for nutrient dispersal, not just horizontally, but in the vertical column also. And therefore, they enhance primary productivity about where in areas where they are or where they moved also. There are major carbon sinks in our oceans, and these 2 points are very important, and they are very much, even grassroots level communities, so fishing communities are very intrigued by these, all this latest literature on these two. They, of course, give ecosystem services, not only in terms of tourism that we are all aware of, but also fisheries management. Samantha, next slide. Especially when it comes to the large whales, like this is an Omora's whale, it's a rare and elusive species, or the dugong on the right, which is also rare and elusive. But the large baleen whales, especially, are indicators to fishermen for shoal presence, the presence of anchovies or sardine shoals. So they can also be used as in that way as a very important ocean health indicator. They are also indicators of water quality, of algal blooms. So when we do have mass trendings of cetaceans, or especially, and if there are proper protocols in place, then they can be an indicator of ocean water quality. And finally, of course, there are indicators, as we have said earlier, the birds also of climate change and of changes in geochemistry in the sea. The next slide. So now that we know why they are important at the ocean level and at the habitat quality level, what is it that we want out of this EOV? So an overarching and a good way of getting a lot of different aspects of marine mammals into the picture is abundance and distribution. And abundance basically refers to the population size, the number of individuals, and distribution is the geographical extent of the space they use across habitat types. And within the abundance and distribution, what are the key measurements that we can use or that will be applicable for GOOS for all the practitioners of who use the data from GOOS? And what can it be used for to observe what, right? So those are the phenomena. We can observe trends by it. We can observe shifts, which are not the usual distribution. There's some kind of change going on. And then we can look at mass mortalities as a way of looking at ocean health. So some of the main sub-variables there are either the presence or absence of dolphins, whales, seals, sea lions, whichever. Then you have count data, and then you have repeated individual presence. So over here in this is what we call sub-variables. These are key measurements, and these are just some examples here. Down in the lower part, you can see the left map that shows just the location. Everything in our key variables comes with location and time and effort. Those are three very important backgrounds. So you can see that you can get simple count data with location and space, and that is also a very good data set. You can go to another level of presence absence, not knowing population size, but at least knowing presence absence using acoustics or genetics like eDNA. Over here on the right, Chico et al.'s paper shows us this is humpback whale data, only based on acoustic recordings in an archipelago in Iceland. And he shows that there is a difference in the way the animals are using the space across seasons. Okay, so the blue part is non-song social songs, so social vocalizations, which is all in the winter, while in the summer it's all songs only. So these are the kind of data sets that can be used. And then a very simple one down with the photographs is photo ID. We can also do that with satellite tagging, of course, where you're tracking animals across space and time. So these are the key measurements that we are looking at. Next. And then we have the supporting variables, as Patricia also explained in the seagrass. We have some supporting variables and that provide scale and context to all the data that we are collecting. Basically, it gives far more depth to the data that we are collecting about we know how many animals are there. Now let's say we know where they are, but what does that mean in the larger picture when it comes to the ocean and everything that's so dynamic in the ocean, right? So let's say we have data on the age class, on the sex, on group sizes, on behavior. All of these tell you about the importance of that particular area that you're looking at. And if that changes, then we have a phenomena that we can observe. When it comes to, let's say, animals that have stranded, like we said, dead, live or dead, absence and presence, then we would want to know what the causes of that are. Or we'd be able to say the body health. The body health would tell you a lot more about, again, ocean condition. If you're looking at estimating only population size, then matters like detection rates and Q rates come into the picture. So these are some of the supporting data sets that we can collect. It would be great if we had it to give more depth to our EOV. Next. And then there are other variables, the kinds that we are more used to talking about as supporting variables, and these are the environmental ones. There's temperature, there is salinity, dissolved oxygen, suspended particulates. I don't need to read out all of these. I think we are used to a lot of them. But in the case of marine mammals, I think, productivity, algal blooms, prey availability, bathymetry, all these are really, really important. And this little diagram on the right by Bestley et al., it's basically showing how different species separate themselves in the water column, based not only on their preference, but also on their physiology, also on their anatomy. That's how evolved marine mammals are. So anything that goes wrong, let's say in a canyon, or there's oil and gas development somewhere, all of these can actually, affect the distribution of animals. So the health of these different habitat types can also be figured out based on our EOV in these using these variables. Next. So what are some of the phenomena that we can observe using our data that we have collected, and how that can then later be used, right? So here is one example which is involving population size and trends, but also in relation to the supporting variable of, let's say, sex. We all know the North Atlantic right whale population. Most of us who are in the marine mammal world know about it, but this is one of those rare small populations that are decreasing. And when we found the paper here by Reed et al. Is explaining that this small decreasing population, why is it doing so? Because the sex ratio, first of all, is much higher of males than females. Okay, and on top of that, when they did a trend analysis of just the females based on mark recapture, they used mark recapture photo ID data, they found that the female population is decreasing since 2010. So that all of that, all these things that we talked about, the EOV, the sub-variable, the supporting variable, all of that has been used to then realize that, oh, this is what is going on with the North Atlantic right whales. So this is one way, one kind of phenomena we can observe using our EOV. Next. Distribution shifts, a very, very important one, I think, to discuss. And again, Patricia also mentioned the same. On the right is a general paper looking at marine species. It's not looking at marine mammals, it's looking at marine species. And the species that that Pinksy et al. Looks at are all ectotherms. They're basically fish that can depend on the outside environment for regulating their temperature. Even then, they have noticed that that these fish species are showing shifts along with climate velocity. Now, what is climate velocity? It's the rate and the direction of change in temperature. So as the temperature is changing, the ectotherms are also shifting. Of course, there are nuances between the different groups of fish as to how they move. But it's a very interesting paper if you want to just look at it, look at an overall picture. When it comes to marine mammals, though, they're an altogether different thing. First of all, they spend all their life in water. They don't have any time outside. Most of them, other than the pinnipeds. And because of that, and they're endotherms, they have to regulate their temperature internally based on their metabolism. So it has been found that they respond slowly to climate, to shifts in climate or sea surface temperatures. And what will be seen, or what has been seen right now, is that there is for cold water species, the range is contracting, while for warm water species, like the species of marine mammals found in the tropics, so to say, the range is expanding. But what that means then in terms of conflicts with other species that were already there in those areas, about sharing prey bases, all of this is something we don't know enough about when these shifts happen. So monitoring them at a global level, at a local level, all of this is very relevant very relevant phenomena to observe, to figure out what's happening at sea. Next. Yeah, finally, it is, the mass mortalities, as again, Patricia has already covered. One of the most recent cases that we are all aware of is the highly pathogenic avian influenza that has happened in elephant seals. And it's a very, very striking example of the fact that these are highly contagious, and that's where One Health comes into picture that I'll talk to you later about. And this has happened even in Antarctica. But very interestingly, the fact that disease in marine mammals is being studied is really, it still happens mostly only in the global north. Where I come from, animals are not even checked for disease most of the times. And we are talking about 2025 now, while this paper by Gulland and Hall, who covered 1996 to 2007, showed that only since I think it was since 1990, that people actually started documenting disease in marine mammals. But given that they're so closely related to humans and the problem of zoonosis, I think this is a very, very important aspect to consider when it comes to phenomena that we can observe and take back and relate it to our sub-variables and supporting variables. Yeah, next slide. So how are we doing all this? How are we able to cover a whole range of questions for marine mammals, given that they spend most of their time at sea? They're elusive species. Some are even rare elusive species like the beaked whales. They rarely come up to surface. The sperm whales can stay down so long that if we were just on a ship passing by, we would probably miss them, right? Unless we know where they are. So some of the methods or approaches that we use as marine mammal biologists is the first one over here on the left. You can see our visual line transact service. That's how we all started, shore-based and visual. And these are some transact lines that you can see over here that are followed and replicates are done. We can also use now towed arrays with this kind of approach because distance sampling requires you not to stop, and that's what towed arrays also want. So this is one way of doing it where you get good density estimates, abundance estimates. The other way, if you are close to shore and you don't have permission problems, you can use a drone to look at group size level supporting variables for your covariables. So, so that is drone-based assessments. And then, of course, with drone-based and remote sensing, also you have to use deep learning and AI to be able to give you accurate data in the end, modeling, of course. Then there is static hydrophone systems, which a lot of us are using now for presence absence data. You just leave the device in the water and you get acoustics, presence, absence of different species that may meet within that area. Of course, there'll be differences at species level as to what the source of the call that can be recorded in your area. While talking about this, I know that Patricia already went through, what the desirable minimum and ideal levels of data collection is. So I think really this would depend on your situation, on your circumstance, on your logistics, on the scale at which you want to do your data collection. We all know that the ideal is that we go out every day and we have a recorder in the water every day, but that's not always feasible. But the minimum is at least something that helps us accurately observe a phenomena, any of the phenomena that I spoke about earlier. The next slide. Some more observing approaches, I think, in the next slide. On the right-hand side, of course, there's satellite tagging, a very, very important approach to take if you have healthy, stable populations. Then there's photo ID that I already talked to you about. This can be done for cetaceans, it can be done for pinnipeds, it can also be done using collars, and it can be done for the large whales that migrate across ocean basins also, Photo ID still remains a thing to be able to look at shifts and movements in marine mammals. Disease assessment we went through. We don't have a very strong database in but I think it is a very important factor. And then finally, but not the least, is citizen science. In my part of the world, again, where we don't have logistics and finances in place, we have citizen science that is giving us distribution data on different species of marine mammals. So this is a website which is an open access database of more than 1,000 reports from our part, from South Asia, Sri Lanka, and India. So I urge you all, if there are such other open access databases, then get them involved, get them in touch with us, and it'll be really great to have everybody providing data into GOOS. Next slide. Yes, so finally, what we all are seeking, and as Sam also explained in one of her slides earlier, is how do we take this all back and make it useful, for different types of practitioners, whether you are an oil and gas person, or whether you are a green energy person, or whether you're looking at ocean health and One Health programs, or you're a fisheries manager, or you're a shipping company. Traffic management is a big thing. So I think once all our databases come together on a platform like GOOS, then all these derived products, they can be used for various derived products like risk assessments at species level, but then spatial mapping for shipping lanes at a larger level. Important marine mammal areas, not only at the country level, but at the ocean basin level. So these are all these, I've highlighted them as some derived products that can be used nationally, regionally, and globally by different stakeholders and practitioners. Once we have all these different data sets and their metadata in one place in an open access format like GOOS. So I think that is where I think I'll probably end. Internet also says it's become slow. Sam, can you hear me? Yeah, so big thank you to everyone, especially to the experts who went through the spec sheets for us and gave their inputs, and to all of you for being here and listening to our talks, and hope we can help you with questions. Yeah, thank you.
Thank you. Thank you, Deepani. Thank you, all of you, for the 3 great presentations. They gave us an impressive landscape on the seabirds and marine mammals EOVs for abundance and distribution. And so we will now start reading the questions from our participants. We have 8 questions, and let's try to answer them all live. So the first 2 questions were put to Samantha when she was presenting, but I think the three of you can give your insights. So the first one comes, and I'm sorry for the participants if I mispronounce the names, but I will try. So the first one comes from Hamed Benarejal. And the question is for Samantha, thank you for the great presentation. Considering the rapid advances in satellite remote sensing, how do you see the integration of satellite-derived environmental data with acoustic monitoring and traditional survey methods improving future assessments of marine mammal abundance, especially in areas experience increasing anthropogenic pressures? So I think also Dipani and Patricia, if you want to give your insights, but yeah.
Thanks for the question, Ahmed. I think you're absolutely right that where we have multiple resources to bring to bear to estimate abundance or distribution shifts or any of our EOVs or phenomena, we'll do a better job. I think most of the challenge at the moment is actually in many areas we don't have even one way of observing the essential ocean variables. So that's where a lot of our capacity building is focused to try and make sure that we have global coverage for these essential ocean variables through at least one medium but you're right that when you can bring multiple to bear, it does definitely improve your ability to monitor through space and time.
So next we have Vasily Egorov. Is it possible to access the data through API or as a WMF server to GIS like ArcGIS or QGIS to analyze it in Python? Samantha.
Great question, Vasily. I'll caveat this by saying that's not my area of expertise, but I believe that OBIS has its own API, and therefore, if you check that out, I can put a link into the chat to the portion in the OBIS manual that talks about data access, and you can look at their API there. Whether or not you can use others, not my area of expertise.
Thank you, Samantha. So next off, we have a question for Patricia from Hamed again. So we have, in regions where long-term monitoring of seabirds is limited, what strategies do you recommend to integrate opportunistic data or citizen science observations into robust abundance and distribution assessments?
Thank you so much for this question. It's really important because we don't have a global coverage of great ideal observation system for seabirds in every colony, but all information matters. So if you do have observation network longer than 5 years, even though it's not as precise as every month counts at the surveying seabirds, the information is precious, it's really important. Of course, we have to access the error estimates on counts and the accuracy of the data. When we do have limited counts of seabirds in a colony, that will make your accuracy of the population size really less precise. But as we do have options as minimal, desirable, and ideal, we can have this different levels of information available. So it's a minimal scale to have the count of the seabirds at a colony every 10 years. That's not quite difficult, but sometimes it's the only thing that we have. It's better than nothing to have that information shared as a minimum for seabird counts at colonies than nothing, of course. But as we do have more effort, more resources to have the ideal, we increase accuracy and precision, so we have more precise trends on the population and species at colony level or island level. So it all depends on the accuracy that we are looking for, and of course will depend on the information available. But any information within the format that is count of nests of mature individuals, it is important in a long term. So thank you so much for your question. That's why we do have levels of option to count, and that the precision will change within the options we have, and all information is important. Thank you so much.
Thank you, Patricia. So we have now a question that can be addressed to maybe Sam, but also I think Patricia and Dipani, if you want to give your insights. But so I'm sorry, I don't know how to really pronunciate the name correctly, but Chandrasekhara Sarma. The question is it is fascinating that EOV data allow us to observe complex biological phenomena. So in your experience, and I guess the three of you can actually put your insight into this, what is the best way to translate these technical phenomena into simple ocean literacy narratives for policymakers and coastal communities? How do we turn EOV data into a compelling story that justifies the creation of new conservation zones? I don't know who wants to start.
I'll go for it. So thank you for the question. I think this is a great question. Exactly one of the touch points that's intended with the specification sheets. The specification sheets are intended to speak to the technical community as well, but also then connect through to stakeholders and to be able to use the information. And I think one of the reasons we've picked the phenomena we have for our essential ocean variables is that they do allow that storytelling capability. So where you have an increase in a population, a recovery that can be a positive story, where you have a decrease in abundance of animals, either due to a mass mortality or a health issue or just for an unknown reason, again, there's a hook there for a story to be told around that change and hopefully engage the conservation community. And for distribution shifts in particular with climate change, where we're seeing animals appearing in areas where they've not been seen before or not been seen for years, again, there's a hook for a story there from the essential ocean variable information being translated through those phenomena.
Thank you, Samantha. I don't know if Patricia or Deepan, you want to add something, or we can move on to the next question.
Yeah, just quickly, I would like to mention that the subvariables that have been chosen are easy to communicate. The population is increasing or decreasing, or are stable. Where they are distributed is harder to measure that in the precision that we need than to communicate, I guess. So there are a lot of tools that we can use to communicate information, and it's important to have good data, precise data, and good distribution of data around the globe to be able to communicate a precise information, more complete information. So I think to communicate those phenomena that we chose is not hard, but it's good to have good data, good metadata to communicate the right information, the more precise information. So I think it's one way that researchers around the world can collaborate to this communication to be more effective as well. But I agree totally with Sam. Thank you.
And Patricia, I just jumping on that, and before going into another question, it's a follow-up question from myself, sorry, but it's a bit related to this. There's like a risk of oversimplifying, the data or the results to make it a bit more easy to communicate to policymakers or communities? So what is the risk of oversimplification, if you know what I mean? Because, it's a complex, trends are complex, and it's,
I think it's a really nice insight, because calculate trends seems easy, but it's not. It has a lot of complexity involved. So when we communicate that to the broader audience, we can communicate that with the accuracy together with that. So we are communicating this information, but we don't have for this species many counts. So maybe that can be communicated as well. When we do have a really high quality data that can be communicated, we can have that highlighted. And when there is some more information needed, we need to communicate that as well. Not only communicate the information we have, but also the detail on that information, how the error rate of that data is available or not, the effort. That things need to be communicated in a simple language as well, but need to be communicated with information about the population trends, for example.
Yeah, thank you, Patricia.
Yes. I wanted to add specific to this question is that when you're working with policymakers and coastal communities, I think before even going into the jargon of trends, I think they really need to know why it's important to have marine mammals or seabirds around in the first place. So how is it actually impacting their livelihood or their health, the health of the water in their villages maybe. So I think the linkages need to come from there as to, at first they need to know that they're part of the nutrient cycle. So, and those aspects are now, it's very important to communicate them. And then if people know how many animals are out there and how it's helping the productivity of the fishing industry, then that really touches them, So if we are looking at ocean literacy for policymakers or coastal communities, then I think we need to have a proper start point for those stories. Yeah.
Yeah.
And even the
Right.
creation of new conservation zones, I think for us in our part of the world, they're more like community managed conservation zones rather than MPAs, because we have a lot of livelihood dependence on the ocean, right? So those are the ways of involving those two stakeholder groups, I think.
Yeah, you're right. Thank you, Deepani. And so again, Hamed is asking also, I think, for the three. One last question. We've seen great examples today of abundance and distribution EOVs. For early career researchers like myself, working in ecosystem transitions, what do you see as the most urgent research gaps where PhD projects could meaningfully contribute to improving bioeco observations? I don't know who wants to take
I'll take a stab. I'm happy to.
Okay.
Thanks again, Ahmed. You've asked a lot of insightful questions today, so really appreciate your participation in the webinar and engaging like this. I'm going to cheat a little bit because perhaps you've discovered working at ecosystem transitions that you often have a lack of data to be able to look at the questions you really want to look at. So rather than it necessarily being a research gap, I'm going to encourage all early career researchers to learn about data standardization and to proactively standardize your data, share it in repositories and make it available for future generations so that you can take advantage of other data sets that people are sharing, and people can take advantage of the data that you probably fought long and hard to go out and collect yourselves. But I think there's tremendous value in that, and that really speaks to that measure once, use many times. And it's really important to have well-documented, standardized data available to answer some of these challenging ecosystem transition or climate change questions that we're facing today. End of soapbox, sorry.
Thank you, Samantha. Great answer. Don't know if anyone wants to say something else, or otherwise I will go to the next question just to try to answer them all. And so we have Johnny Conjarla that is asking for Deepani. All these variables are essential for all species from mammalia, or it will vary from species to species?
I think if you're talking about marine mammals and the supporting variables or the subvariables, then yeah, I think all of these are the ones that we want to at least have the minimum ones, the key measurements for most of the marine mammals, if that's what he's asking. Yeah.
Sure. Okay.
Is that Samantha?
Yeah, I don't know. I think so. I think this was the question, like if these are like common for all, or if we have like species specific.
There'll be some difference in, like, if you look at the pinnipeds, there'll be some differences in the way measurements are taken compared to, let's say, the large whales or compared to the small cetaceans. Yeah. So the way measurements taken will differ. Perhaps even the methods will, the accuracy of what you are collecting will also differ. Like in seals, you can actually see all the individuals. But if you're looking at dolphins, then most of the animals are probably underwater. So your count methods are far more nuanced in that case and longer, I would think, than seals that are hauled outside, no? So such species level differences, yeah, they will be there. Sam, you want to add?
Yeah. Thank you.
Say the same.
Yep. Okay. So Lindsay Martinez has put a question during the Deepani presentation, but I think it's also it covers a bit the three of you. So to measure all these variables, I also had this question a bit similar. Is it necessary to use like a standardized protocol? Any protocol is valid, and to measure all these variables, do we need to use this standardized protocol? So what is the level of the level of the data, the organization that you need? Is there a template, or is it a bit more flexible, and how this works to include this data into the EOVs assessments? I think this is the question.
So go for it, Deepani. You want to answer?
No, you go ahead. I can add.
Okay, I was going to say two things. I think within the specification sheets for the marine mammals, we provide links to some of the protocols that we're currently recognizing or kind of putting forth. We're not fully endorsing yet because we haven't engaged fully with the community to kind of put forward those endorsed best practices or standard operating procedures. But secondly, any protocol, if it's well documented within the metadata that you're then sharing, is valid because people will be able to understand what you did, how you collected your data. So two sides to that equation. If you're not using one that might be currently put forth by GOOS, then making sure you're documenting what you have done is the key part.
Yeah, I'm sorry.
So,
Yeah, sorry, I didn't like go into the details of the spec sheets, but if you do go there, then we have provided for each of the approaches, we have provided at least 2 references as to the the ways the so-called standard protocols that most people use for that particular kind of approach. Yeah, so you can look at that via the links that were shared. Yeah.
So in summary, if you can use the ones that are provided, if not, please save your metadata to inform on your datasets, but do not stop from sharing them. Okay, so then we have a question from an anonymous participant for Dipani, and the question is, are some pinniped breeding aggregation properties important to observe for the marine mammal EOV?
I'll pass that on to Sam. I'm sure she has lots to say.
Okay.
Dipani
Yeah,
works with
maybe.
cetaceans. I work with pinnipeds,
Yeah,
so I get this
yeah,
one, I
you're
guess.
right. So maybe this one is better for you.
Yeah. So I think very quickly, any of those supporting variables or properties of the breeding aggregation that might influence the EOV, so the abundance, probably in this case only, are the ones that are important to record and try and have with your observations.
Okay. So throughout this discussion, we had a bit more questions. I will read the 3 ones that are left, and then maybe we need to stop by then. So if you then have further questions, I think that our participants left their contact, so you can then put the questions after. So Kathy Vignes Raposa made a question for the 3 of you. Yeah. Sorry, I don't know if I missed. No, it's that. So I feel that biodiversity has gotten to be a buzzword that captures many concepts. Could you please provide your definition of biodiversity? This is complex. So I don't know who wants to start. Yeah.
Okay, I'll go ahead, Dipani. Yes.
Yeah, I think I understand where Kathy is coming from. Like sustainability is a buzzword also. Once upon a time, climate change was a buzzword, by the way. So, so I think for me, biodiversity is just I don't know. It's the entire biomass of life that lives in water, in air, in soil, in earth. Biodiversity is a whole range of species that can and cannot be seen summed up together. Now we know that even rocks have life in them. So biodiversity is that vast expanse of all living organisms connected to each other. It's not a scientific definition. I'm sorry. I'm just giving it from the top of my head, but Sam might be better at that. Yeah.
No, I was going to agree with you. I think originally for me, biodiversity coming through classes, school, university, or whatever else was species richness, and it was limited to that. And for me now, it's much broader than that and refers to the diversity of all biological life on Earth. So kind of as you said, Dipani, and that probably doesn't help with the buzzword mantle, but that's what it is for me.
Great. So João Bernardo Barreiros says, thank you all for your respective presentations. And my question is, with the growing role of tourism, specifically with cetaceans, what are the pros and cons of their potential contribution in research that you've experienced? Did you feel acceptance from the industry, an opportunity to collect more data, maybe also to approach locals or policymakers through an economic value perspective? Thank you for your availability. Yeah, great question, João.
I can go first and then Sam, you can, yeah. I think it's a very, very good question. again, I'm sad to say that the answer to this differs from place to place and even between the global north and the global south, as they say. In some parts of the world, tourism is completely unmanaged. In other parts of the world, tourism is actually well managed and is helping the scientific community. In my case, I have experienced that the industry, if it is the way it works in India, is there are too many people doing tourism at the same time, and so it actually has a negative impact on the species rather than a positive feedback loop into science and into conservation funding and all those matters. Also, in many parts of South Asia, we don't even have a tourism industry in the sense of tourism ministry to manage or have policy that can then govern or limit or monitor tourism activities. Yeah. So even though tourism could contribute to research as being platforms of opportunity to collect data. We have still not reached that stage where I come from. But I think Sam might have a more positive thing to say, I think, perhaps.
I think it's a balance, Dipani, as well. And I think even in parts of the world, in Hawaii, ecotourism interactions with spinner dolphins has been detrimental to spinner dolphins there in the resting bays in Hawaii. And that's with some existing programs to try and promote eco-friendly tours there. So I think as the question kind of framed, there are pros and cons. And there maybe has been in some places some opening of the doors to researchers being on those vessels to be able to make the observations you mentioned, Dipani, so platforms of observation, but certainly the con is the risk of disturbance to the marine mammals and impacting the marine mammals through that activity. So it is finding a balance between those pros and cons.
Yeah. And final, the last question that I will put from the Q&A box, on Dipani's point of communication prior to the study, are any co-designing approaches included in these studies?
I'm not sure I got that question, but perhaps Chandrasekhara might be referring to working with fishing communities before starting a project or a research, a program in a particular area.
Mm-hmm.
That case, I wouldn't say that it is so planned and so strategic. I think a lot of stuff when it comes to working with fishing communities is about first getting an idea of their feel of the project and then changing the project in ways that it is not seen as a hindrance to them or a threat to them. So yeah, there is some amount of time before a project is initiated so that all parties involved, because in my part of the world, we don't drive our own boat, fishermen drive the boat for us. So if they don't want to go on line transects, they're not going to go on line transects for you. So becoming a team first before starting a project and communicating in such a way where everyone's happy with what is going to be carried out. Yeah, I think that is important. And same is the case if they need to know beforehand if what we collect, the data we collect might lead to some changes in designation of fishing areas. So that's a very, very important part also. Should be allowed, if they find out they will not be allowed to fish anymore, they may not want to be part of your research project. Yeah, so everything,
connection goes a long way, right?
everything has to be out in the open from the start. Otherwise,
yeah. Thank you, Dipani. Just to finish off, I would like to make a last question for the three of you, just to close the seminar. Given the global changes that we are facing in such a rapid, way, and the many gaps that we still have in terms of monitoring, do you think that the monitoring techniques and the data availability are becoming are coming to us fast enough and developing fast enough to answer these data gaps in time to correctly address the challenges that we have ahead? So it's a bit more insightful.
An easy question to finish with, then, Mafalda, is what you I think in many instances we're finding new ways to use existing technologies. We're developing new technologies. I think environmental DNA is promising on a lot of fronts.
Yeah.
The cutting edge with marine mammals at the moment is trying to observe them from space using very high resolution satellite imagery. So I think there are a lot of these options and possibilities. And AI offers untapped potential to this point, I think. So in terms of analyzing these ever increasing large digital data sets that we're now able to collect, whether or not it's fast enough, we'll probably need bright early career researchers like Ahmed out there on the front lines in the coming years to make sure that happens.
Yes. So there are no more questions, and I think we are past beyond the time, 20 minutes beyond the time. But so I would like to thank everyone that stayed until the end and that were with us until the end, because it was a really, really interesting webinar, at least for myself. And thank you all three and all the audience for being on the other side and actively participating in this discussion. Thank you all for supporting another Marine Biodiversity Networking Friday. Please keep on following and participating in these discussions that are truly construction bricks for the understanding and protection of our ocean's health. And I think we're done. I wish you all a pleasant weekend. Thank you very much. And I would say that the session is now concluded. Thank you.
Thank you.
Thanks, everybody.
Bye-bye.
Thank you. Bye-bye. Thank you.
Bye.